I'm curious. I am curious. Curious.
By nature.
Welcome to Curious by Nature. I'm Jessica Johnson from Newswise. Vision is one of the most important senses we have. The loss of vision is tragic and has horrible consequences. Our guest today is working to solve blindness caused by macular degeneration, by aging and disease. It's fascinating how he's using stem cells to solve this problem. Dr. Bernier, welcome. Please introduce yourself.
So my name is, I can say in French, Gilbert Bernier, or what is it in English? Gilbert Bernier. I'm a professor at the University of Montreal in the Department of Neuroscience. I'm curious about embryonic development and how you can use this knowledge to cure human disease. So my main idea was to use all this knowledge, this basic research knowledge, to now tackle human pluripotent stem cell to become retinal cells, eye cells, and to cure blindness. So that was the origin of this whole research program. So I started working with human embryonic stem cells in 2004, 2005. And the idea behind working with those cells, probably you know about that, those cells are derived from primary human embryos, so very primitive. And they are pluripotent. So technically, they can generate any cell type of the human body.
So can I just ask, you said they're embryonic stem cells.
Yeah, I'm working with both, also with induced pluripotent stem cells. So human embryonic stem cells are derived from in vitro fertilized. So you're not killing baby. Actually, you do in vitro fertilization. So you take oocyte sperm, you do it in the dish. You do a fertilization, and when the embryo is 64 to 128 cells, you just dissociate the cells. And if you're good enough, you can derive a cell line, which is called a human embryonic stem cell line. In the US, but also elsewhere in the world, there was some ethical concern about working with human embryonic stem cells. It's not the case anymore, but they remain the gold standard in the field, okay? But then in 2007, 2008, a fantastic Japanese researcher, Shinya Yamanaka, discovered actually by himself a method to reprogram somatic cells into the equivalent of human embryonic stem cells, which he called induced pluripotent stem cells. So what are somatic cells? Somatic cells are the cells you find normally in the adult body. So it could be skin cells, any cell type that still can divide. If you do a biopsy, for example, you can do it from blood. So actually, about any cell type that has some proliferation potential of your body, I could derive with this method induced pluripotent stem cells from your cells. So that was a very big discovery, and he earned the Nobel Prize of Medicine in 2012 for this discovery.
I mean, really, I think that the general public hears stem cells and it is not necessarily clear about maybe the origination and there was a period of time when women were saving their umbilical cord from their pregnancy, right? And in order to have those stem cells for future use.
This is a fantastic question. Actually, I'm the one giving the course for the past 20 years at the University of Montreal about cell therapy and what are stem cells, and it's very complicated actually, because depending on the developmental stage, so you really start from the primitive embryo toward the fetus, toward the newborn, toward the adult, you will find a broad range of different stem cells, okay? And the principle is very simple. As the body matures, so the embryo throughout the fetus and then the newborn to the adult, you still have stem cells, but they get more and more specialized and they lose their pluripotency. They become either multipotent or even unipotent. So to give a very simple example, we have lots of stem cells in our skin, okay? In the hair follicle, there's a population of stem cells which allow regeneration of our skin throughout the life. Okay, our skin is entirely regenerated. Those cells are stem cells, real stem cells, adult stem cells, and they can generate basically all the cell type. There are multiple actually you find in the human skin. So these are adult skin stem cells, and they are multipotent, not pluripotent. So a lot of researchers are working with adult multipotent stem cells. When you do a bone marrow transplantation, you're isolating blood stem cells, which you can find in the bone marrow, and which have the ability to sustain the entire blood regeneration throughout life.
That's very helpful to have a basic understanding. Okay, so let's maybe get into your research paper that was in the journal Development. And can you tell me more about that publication?
Actually, the first publication was also in development in 2015. Well, I developed myself a method to give the information to pluripotent stem cells to become a human embryonic retina in the dish. Basically, that's the thing. So, and this method is very well described in the first paper, 2015, actually using human embryonic stem cells, where you can generate millions and millions of retinal cells, a lot being photoreceptor cells, about 50%. And what is very unique about this method is that it gives really a flat, it's a flat retina growing in the dish. And actually you can have tens of dish like this, so it's the factory. That's the idea behind using pluripotent stem cells. They're infinite capacity of proliferation when they are undifferentiated. Okay, so you can keep them in the lab, in the incubator, undifferentiated. You can expand them millions of times, so you can really build the factory. It's really the big potential of those cells is that they can generate any cell type, but also if you maintain them undifferentiated, you can generate millions and millions and millions of them. So if you want to think about the human body or human organ, they're very big.
Are there factories of cells right now? Are there companies that are doing that?
Oh yeah. Oh yeah. It's a big field. Cell therapy is a big field. So in this first paper, we developed this method. We find one molecule. We discovered this molecule which has the capacity to force the cells to become retinal cells, and mostly actually cone photoreceptors. So now I'm getting more specialized, but cone photoreceptors are the cells which have the capacity to cap light. So their response to photons. And in the human retina, there's two types of photoreceptor, rods and cones. Okay. The rods are for night vision and peripheral vision. So it's black, gray vision, white and black vision, gray vision, and night vision. They're very sensitive to light, so they're only activated with dim light. So, and that's a huge part of the human retina is composed of rods. But we also have cone photoreceptor, which actually their response to intense light, and they are the one allowing us to see the visual spectrum of color. Okay, it's called cone photoreceptors. So actually, we're talking together. You're seeing me in the camera. It's only your cones actually operating in this process. The rods are sleeping. It's too much light. And what is specialized in the human retina, but also in great ape, is that we have a structure which is called the macula. So everybody knows about macular degeneration, age rate. I mean, something that everybody's hearing frequently. Well, this structure is at this more or less the center of our retina, and actually that's the structure that is highly enriched for cone photoreceptors. And that's the structure that is mostly involved when we're reading a book, when we're looking at somebody, talking to somebody. That's allow us to see the face. And also it's involved in very high resolution image. So the human eye has a fantastic capacity of resolution. That means we can see very small thing or distinguish two different small thing. Okay. So this structure is also involved in this process. It's very, very refined, but the side effect is that it's a very sensitive structure, and it's frequently affected in either acquired disorder like age-related macular degeneration or genetic disorder. So inherited disorder, people will lose vision and central vision either as child or in the adulthood, and they become blind. So they are legally blind because their macula is completely degenerated. Okay, so it's a big problem for human vision is the loss of macular function. If you lose the structure, well, you have a big black hole in the middle of everything you're looking for. So people which suffer from loss of macular function, actually, when they talk to you like this. Huh? They try to see you with the side of the eye because they don't. The central vision is lost, completely lost. So the whole idea behind this project: can you generate something similar or close to a human macula in a dish, in a cell culture dish, using human pluripotent stem cells? And that's actually, I think that's what we achieved to do. So we generated a human embryonic retina, but that is highly enriched for cone photoreceptors. And we believe that this patch, which we call, and maybe be used as a tissue replacement therapy to actually put a new macula in the human eye. Okay, so the paper we published in 2024 actually is the experimental test and demonstration of this. So again, using the same technology developed before, this time using induced pluripotent stem cells. I mean, it's not very important why we use induced pluripotent stem cells, but in this article, we use those cells, which are the equivalent of human embryonic stem cells. And so what we did, we wanted to use a large animal model of retinal degeneration. The main goal of this research is to restore vision, to cure blindness. So that's what we did. We did that in pigs, which are very big, and which also have big eyes. It's very important because their physiology is very close to that of humans, their size, but also the size of the eye is very large, and that allow a human retinal surgeon, so a real surgeon, which would operate me or you, to do all the surgeries. And it's not a scientist like me, just it's a real human retinal surgeon that did all the surgeries in those animals because they're big enough that it's very similar to the human eye.
What is the difference between a human eye and a mini pig eye, which was used in your research study?
It's a bit smaller than the human eye. So it's about 70% the size. But otherwise, it's almost identical, except they don't have a macula like humans. So what we did, we use a drug which actually destroy all the photoreceptor from a region of their eye. So we generate kind of a macular degeneration, although they don't have a macula. So in the region which has both rods and cones. So we generate this region which has no photoreceptor anymore. We waited for 1 month. We did all kind of functional tests with sophisticated machine to actually confirm that there was no visual response to bright light, so daylight in this region. And once this was confirmed, now we did the surgery, very complicated surgery, to put this human retinal tissue, which is basically like a pancake, So it's about 4 millimeter in diameter, so it's circular, but it's very thin, so 100 micron thin. So it's a big pancake in terms of biology. And now we had to put this pancake at the right place, so where we generate the lesion. Okay, that was actually the most difficult part of this whole project. But when it succeeded, what we found is that after 2 months, now in those animals, we could see now recovery of function, not completely, but still from complete absence of function in this region. Suddenly we could see spike. So when we pulse the eye with bright light, we could see a response from actually which are coming from cone photoreceptors. So these human cone photoreceptor in those pancake which we put inside, they could respond to light and actually transmit this signal to the retina, to the rest of the retina.
What's happening next? Are you continuing to work on the same issue, or are you doing something else?
Oh no, I'm I want to get this thing into the clinic. That there's no, I mean that's my goal. I mean there's no nothing's gonna prevent me to do that. Uh, so there's another important thing we found in this article is that it's safe. We haven't we grafted 24 animal both eyes, so it's a lot of transplantation, and we haven't see any adverse effects. So people are very scared about possibility of tumor formation and stuff like that. When you do this kind of transplantation, actually it did not happen. So it was very safe. We did also safety study in nude mice, so mice which don't have an immune system, and actually nothing happened when we graft those cells. So that was an important part of the study. What is the next step? Next step is to grow these retinal. We call them retinal sheets because we have to give them name. Or these human macula grow in the dish on a support. So we're building semi-rigid support, which have been tested in human before, and they will be grown on that. So and these support can be fold. So the idea now is to again going back to the pig, but uh, place the retinal sheet on the support and make again the surgeries. And our expectation is now that we should have a very high rate of success of placing the tissue at the right place, but also preventing folding of the retinal sheet. And if we succeed doing that, would be ready to go in phase 1 clinical trial. So
How many people are affected by macular degeneration?
macular degeneration, so the age-related macular degeneration, or it's a huge problem in Canada, in the US, and in developed countries. We're talking about millions of people, basically. Number are very large. But also beyond those people, there's also quite a lot of people suffering from inherited retinal disorder. People which are losing vision as a child, 5 or 10-year-old, or even as an adult, 20-year-old. And we're not hearing much about those people because they don't have a strong voice. And another problem is that those disorders are very, there's multiple disease, genetic disease leading to blindness. So it's not a single disease, it's multiple disease. So all those are kind of orphan disorder, but it's hundred of different orphan disorder leading to the same endpoint, which is blindness. And in Canada, we're talking about we did some estimate. We could treat 10,000 people per year continuously just for inherited retinal disorders.
Why did you decide to research vision and solutions for macular degeneration?
I would say it's by chance. Initially, I wanted to work on cancer. That was my idea when I started graduate school. But then finally, I did my PhD on neurological disorder, so neurodegeneration, with my mentor at that time. And actually, I became fascinated by neurological disorder. So no, not cancer, although cancer is also very interesting. And then again, a bit by chance, I moved to Max Planck Institute where they were doing developmental biology, and my PI say, "Do you like to work on retinal development, eye development? How do you build an eye from scratch?" I said, "Well, this is fantastic." So they had multiple mouse mutant for all different kind of eye development defects. So no eyes, and ophthalmia. I mean, and then we started to play with the genetic of how do you build an eye, from scratch.
Yeah.
And then, so when I finished my postdoc in Germany, I say, well, now that we know pretty much how to build an eye from an embryo, well, why we cannot use this knowledge now to build a retina from human embryonic stem cells to cure people with those disorders? So really, everything just, uh, follow up naturally. And that's where I am today.
Thank you very much, Dr. Bernier, for your interview.
Curious by Nature is a production of Newswise. More than 7,000 journalists get the Newswise wires. Visit newswise.com and find out how to spread your news today.